Hi, this time I'm going to look at the Single Award Science Unit 4. That's the practical unit, and this is Booklet B, so the written paper, and this time it's the higher tier paper. So this one there's an hour and 15 minutes for. It's still 70 marks, but slightly longer than the foundation tier paper. So here we go. Section A is the biology section, and it starts by asking you to describe how you would make a temporary mount of plant cells for observation under a microscope. Your answer should describe how to prepare a slide containing plant cells, for example onion cells, and how to focus the microscope so that the cells can be seen in detail. So this question also appeared on the foundation tier paper, the specimen stuff as well. So it's one of these communication of specialist scientific terms, communication skills questions. Okay, so the indicative points that they gave in the mark scheme were this. So first of all, you'd remove a section of epidermal tissue from an onion, it said, or other tissue, plant tissue. So it said, for example, an onion cell up here, so it didn't have to be onion. The epidermal tissue is the little layer of sort of skin in between the layers of the onion. So if you described, peeling the layers apart and taking that layer of skin off, that would be a good way of talking about removing a section of that if you didn't remember the word epidermal tissue. So then the second stage is to place that onto a slide, adding iodine or water. And then the third thing you would need to do is to add a cover slip, and then put the slide on the stage of the microscope, and then move the lens or the stage away from each other, and then focus at low power first, and then focus at higher power. So if you're hitting 6 to 7 of those points, you'd be into the 5 to 6 mark bracket, and with your good communication skills, that would give you 6 marks. 3 to 5 points, you're in the 3 to 4 mark bracket, and again, communication skills would get you 4. And then for 1 to 2 points, you're in the 1 to 2 mark bracket, so probably 2 marks, assuming you've written your answer in coherent sentences and used specialist terms. Okay, part 2. When viewing onion cells under a microscope, a student noticed a number of dark rings in their preparation caused by air bubbles. Describe how these air bubbles could be prevented. Now, not fixed, but prevented. So that's prevented, by lowering the cover slip at an angle or using a mounted needle to lower the cover slip. 1 mark for saying that. Then the diagram shows an onion cell that was observed by one student. Other surrounding cells are not included. So on the diagram above, label parts X and Y, so ignore everything else at the minute. So X is the cell wall and Y is the nucleus here. Then it says plant cells also contain other structures that are not shown in the diagram above. Add and label 2 of these structures to the diagram above. So you'll see here I've labelled the vacuole, here the gap in the middle, I've drawn it on. The cell membrane is just inside the cell wall, and then the cytoplasm is just this stuff around the rest of the cell. Now, it is conceivable that you could have labelled chloroplasts on there. I would say that steer clear of that if you're talking about an onion cell, because onions grow underground, so they're unlikely to have chloroplasts in them because they don't photosynthesize. If it was a leaf cell, yes, absolutely, but it's not. Next part of that question, the students counted the number of cells they could see in their field of view at high power. Cell detail is not included. So some of their results are shown in the table below, and you see here we've A, B, C, D and E's, different students, and then the number of cells they've counted are shown, and the total is 83. So it says calculate the number of cells counted by student E, which is blank in the table. So basically the total is 83, and then we're taking away all those other numbers. So 83, take away 23, and 23 and 7 and 7. So that actually leaves you with an answer of 23. And it says one of the reasons the students counted different numbers of cells was that they were confused by cells that were only partially in the field of view. Some of the students counted these cells and others did not. Apart from either deciding to include all these cells or to exclude all these cells, give 2 other approaches the students could have taken. So I suppose one of the approaches would be to count all the full cells and count the half cells and add them together. So they're assuming that, you're taking like anything that looks like half a cell, so here, so you would count that as 1, 2, 3, and so on. Another approach that they could have taken would be to add the partial cells and okay, I think adding, like, add up. I'm just thinking how you can word this. Add up the small parts and then the full cells. So I think that's probably sort of a sensible way to think about it. So you're taking little bits and adding them together to make up a full cell. Okay, so 2 marks there. I think anything sensible really is going to get you a mark in a question where it says, give to approaches or suggest something. So then animal cells can be observed under the microscope. When counting animal cells, the student noted that there were many more animal cells in the field of view compared to plant cells, so more animal cells than plant cells. What does this tell you about the relative size of animal cells compared to plant cells? It tells you animal cells are smaller than plant cells. And give one thing that had to be done to ensure that any comparison between plant and animal cells was valid. Well, you need to make sure you're using the same magnification or you're looking at them through the same lens. Number 2, Benedict's reagent is the test for reducing sugar. If enough reducing sugar is present in a sample, the reagent will change from blue through green, yellow, orange to a brick red precipitate. So apart from adding Benedict's reagent, what else needs to be done when carrying out this test? You need to put it in a water bath to heat it up. What's meant by the term precipitate? So it's a solid that forms in a solution. So when you add something to a solution and you get a solid appearing, that's a precipitate. Now, a student tested 3 foods with Benedict's. All other variables were controlled, for example, the mass of the food, the volume of the reagent, and her results are shown in the table below. So food A had the most sugar. Explain the results in the table above. So A had the most sugar, and you can see that because it went from blue to brick red. And then B and C had the same amount of sugar. Why? Because they're both orange. Okay, so we've got the food, the colour at the start, the colour at the end. If all 3 foods had turned brick red after testing with Benedict, suggest how the student could have distinguished between the amounts of sugar in each food. So you know that you have to heat it up and watch it change colour. So if you recorded the time it took for each sample to go brick red, that would be a measure of how much sugar was in each sample. So then describe how to carry out the test for protein. So we're looking at adding the Biuret reagent to the food sample and then adding a drop of copper sulfate and shaking it. Didn't say anywhere in the mark scheme about the result for the test because it's just describing how to carry it out. But I think it would be valid to say that it goes from blue to a violet colour at the end if protein is present. Okay, question 3. This is one that occurred on the foundation tier paper as well. So when an alkali is added to an acid, a reaction referred to as neutralisation occurs. If in this reaction, an acid and an alkali form a salt and water. When a person is suffering from indigestion, hydrochloric acid is collecting in their stomach. Indigestion remedies contain an alkali which neutralise the acid, leaving the person feeling better. The student wants to determine what volume of alkali would be required in a bottle of indigestion remedy in order to neutralise the typical concentration of hydrochloric acid found in the stomach. The student is provided with a suitable concentration of hydrochloric acid. Describe how the student would carry out the investigation to find out how much alkali would be needed to neutralise the hydrochloric acid. A burette attached to a stand, beakers and graduated cylinders are provided. So you should start by drawing a labelled diagram. So quite a lot to read there. When you get a blurb like this, make sure you read it all first. Don't just go straight down to the question here and think, right, what's it asking? And then kind of hope back through it. Like, you need to treat it like you would a comprehension in English. So read the whole question first, okay? So it tells you about the burette attached to a stand. So that's the first thing you draw. There's the burette. And then you need to have a beaker with acid in it as well. So 1 mark for each of those being drawn and labelled. And then to actually carry out the experiment, we're talking about neutralisation. So you're just adding the alkali to the acid, you're recording the pH, and then giving understanding that the neutralisation happens when the pH is 7. Now, it actually went on to the next page for this. There were 5 marks, okay? And those 5 marks were 2 from the diagram and 3 from your description. Now, part B, the graph below was plotted by the student when they completed collecting their evidence. And you see here it's got the volume of alkali added, and it's got a curve on it with quite a steep section in the middle. It says complete the labelling of the y-axis in the graph above. So when you look at the y-axis, y of course is the one that goes up. That's why we tell you to remember y's up for the y-axis. It goes from 0 to 14, so it's always a good assumption that if something goes from 0 to 14, it's pH, and we know it's neutralisation, so you're labelling pH there. Then what volume of alkali should be added to the bottle of indigestion remedy? So this section of the graph here is where pH 7 comes in, so it's neutral, so that would be 100, so 100 cubic centimetres. Your explanation is that's needed to neutralise the acid. Okay. Part C, tooth decay is caused by acid in the mouth corroding tooth enamel. Toothpaste is usually alkaline to neutralise the acid in the mouth. Describe how you could use an indicator to identify the pH of the toothpaste. So you would dip it into the toothpaste and then compare the colour it goes to a colour chart. 2 marks for that. I suppose you could have talked about using a liquid indicator instead, but still would need to compare it to a colour chart or to a table. So the pH of the toothpaste can also be measured more accurately using a pH probe. Give 2 reasons why. So the probe measures to 1 decimal place, so that means it's more accurate than just saying it's pH 1 or 2 or 3. You can say exactly what it is. And the other thing is that indicators are subject to interpretation. So when you look at an indicator, you're comparing it to a chart or a table, trying to work out, is it orange? Is it a yellowy orange? Is it not more of a reddy orange? What type of an orange is it? Okay, question 4. The reactivity of metals can be compared by observing how they react with water or acid. Name the gas produced when metals react with water or acid. So, hydrogen. Describe fully how you can test for this gas. So we're looking then at saying, we know that it gives a squeaky pop, but it says describe fully. So 2 marks here, so we need to say 2 things. So the first thing is put a lit splint into the gas, and then it burns with a popping sound as your second mark. Then the table describes some observations when metals react with water. We have sodium, which moves on the water surface and disappears. Potassium burns with a lilac flame, moves quickly on the water surface and disappears quickly. Copper, which just sinks, and calcium, which sinks, and then after a period of time starts to rise and fall, producing bubbles. Place the metals in order of reactivity. So the most reactive is the one which is doing all the quick stuff. So that's potassium. Then sodium, which is reacting on the surface of the water. Calcium, which sinks and then starts to react after a period of time. And then good old copper that just sinks and does nothing. All of those correct got you your 2 marks, and any 2 in the correct sequence would have just got you 1 mark. So if you made an error but got 2 in the correct sequence, that was 1 mark. One thing that had to be done to make sure the comparisons were valid would be to use the same amount of metal. And how would the reaction of the metals change, if at all, if acid was used instead of water? So we know that acid reacts more vigorously, so just more reactive. Scientists investigated the complete combustion of an alkane in oxygen. The complete combustion of an alkane can be represented by alkane plus oxygen gives you carbon dioxide plus water. So they measured the volume of carbon dioxide produced over time, and the results are shown in the table below. Now, it doesn't matter if you've never done that experiment before, this is testing your ability to understand this table of results. So we've got time in minutes and then the volume of carbon dioxide produced in cubic centimetres. And it says calculate the average rate of reaction over the 10 minutes. So at 10 minutes, you've got 17.5 cubic centimetres produced. So we do 17.5 divide by 10 to give us how much is produced in 1 minute. So it's 1.75 cubic centimetres per minute. So 1 mark for the working out and 1 mark for the correct answer. Then the scientists repeated the investigation but doubled the amount of alkane used, assuming the volume of oxygen available is in excess, that means there's more of it than you need, suggests the effect that this will have on the rate of reaction. So if you're doubling the amount of the alkane, you would suggest that the rate would increase. You could also have suggested that it might double. Now we're on to section C, which is the physics section, and it starts off with question 5, talking about the diagram showing how the speeds of reaction can be investigated by 2 students. We've got A and B, and you can see probably a very familiar image of a ruler, and you know that the ruler would be dropped, and that's how you measure reaction time. So we've got A here and B here. It says, using the information provided, describe fully how this equipment can be used to find which student has faster reactions. So I'm going to start by saying A holds the ruler and drops it, and then B catches the ruler as quickly as they can. Then you record how far down the ruler it is caught, and you see that in this picture here, and that indicates the reaction time. And then you repeat for student B dropping the ruler and A catching it, or you could have, written it the other way around to begin with. But those are the 4 points for 4 marks there. Now it says reaction times can also be compared digitally by individuals pressing a buzzer when they see a particular shape flash on a screen. Two students had several attempts at measuring the reaction time by this method, and their results are as shown in the table below. So we have M and J, and they have 5 attempts each, and it asks you to summarise the results shown in the table above. So when you look at M, 0.44, 0.41, 0.36, 0.35, 0.36, and then J, 0.67, 61, 55, 77, and 45. M's reaction time is always shorter than J's. You can say that to begin with. And then both students' reaction times decrease with time initially. So you see at the beginning here, they're going down certainly for the first 3 attempts. Okay. M's then levels off after 3 goes. So we see the last 2 for M are sort of similar, but J's keeps decreasing. Now, when we look at this one here, the 4th attempt is an anomaly. So if you said J's 4th attempt didn't fit or something went wrong with J's 4th attempt, that's your 4th mark there. Now we have a circuit diagram, and it says the circuit was connected by a student who was investigating the relationship between current and voltage for a length of resistance wire. Using the letters A and V, label the ammeter and voltmeter in the circuit. So the ammeter is the one which is in series, and then the voltmeter would be in parallel with the wire. 2 marks for those. Describe the arrangement of the components in this circuit. So the ammeter is in series with the battery and the variable resistor, and the voltmeter then is in parallel. So 2 marks there. Then describe and explain the function of the variable resistor in this circuit. So the variable resistor as a slider, that's describing it slides, and then its function is to change the length of resistance wire in the circuit, and when you have more resistance wire in the circuit, that lowers the current. So something like a dimmer switch on a light, or maybe a volume control, would have a variable resistor in it. So the student recorded the values of current and voltage displayed on the meters in the table below. On the grid below, plot and draw a line graph of these results. So you're just plotting the points on. All points correct would be 2 marks. 2 to 4 points is 1 mark. And then a best fit line is another mark. Now, my feeling is that a best fit curve here is the best. It doesn't say a straight line graph, so that is fine. Now state the trend of your graph. So going back here, you see it's going up. So as the current increases, the voltage increases. And it's only 1 mark, so you're not having to give values there with your answer. Then when the voltage across the bulb is 1.5 volts, the current is 4.5 amps. So use the equation voltage equals current times resistance to calculate the resistance of the wire. So current the voltage of 1.5. So we'll have to rearrange this, don't we? So the voltage is the current times the resistance. So resistance is V over I. Okay, so that's where this is coming from, the voltage divided by the current, which is 0.33. 1.5 divided by 4.5 is 0.33. And 2 marks for that. 1 for showing this part of the calculation, and 1 for actually getting it right. Then, using the information provided, state 2 things that were done to ensure the investigation was a fair test. So the 2 things were, it was the same length of wire, and the other thing is the same battery. You could also have said the same voltmeter and ammeter each time. And that was worth 2 marks. So any 2 of those would have got 2 points for you. So that is the end of the higher tier paper. You see there are some similarities with the foundation tier. Now, if you haven't watched it, it might be a good idea to have a look at it too, because there's a couple of experiments in it which don't feature in